Welcome back to the lecture series in animal physiology. So, into the third class of the 12th week. So, today we will talk about blood. So, we have talked about the cardiovascular system, respiratory system. Now, today we will talk about the fluid which is actually flowing through these blood vessels. So, about blood, as we always say, as long as you are not seeing it, it is essentially out of sight and out of mind. You really do not bother much about it. So, the way you can visualize blood is basically a fluid suspended with lot of proteins and other biomolecules and a bunch of different kind of cells, and which could be classified as the largest number of cells are called the RBCs, red blood cells. They are red in color because they have a molecule called hemoglobin, which is a porphyrin ring. On the porphyrin center there is an iron sitting with +2 oxidation state, and this porphyrin ring along with the iron imparts it a red color, and that is why it is called a hemoglobin. And very closest relative of it is chlorophyll, where iron is being replaced by magnesium, and it, because of the presence of the magnesium, it gets a green color. Okay. So, these are some of the very fundamental things about it. So, the hemoglobin is involved, and we will talk a little bit more about hemoglobin as we will scroll through the slides about how hemoglobin binds to oxygen and at different spots where it get rid of the oxygen and continue its circulation. Okay. So, talking about the blood. So, let us move on to our first slide. So, the topics what we will be covering is introduction to blood, function of blood, composition of blood, then we will talk about plasma, formed element and homeostasis. This is what we are going to cover. So, introduction, the basic component of the cardiovascular system, which is circulating fluid, which is the pump. We talked about conducting pipes, we have talked about it. The conducting pipes includes your blood vessels. Coming to the functions, it transport gases, nutrients, hormones and carries the metabolic waste to be discarded from the body. It helps in pH regulation, maintaining osmolarity, and this osmolarity value, it varies around 320 milliosmoles. Okay. Restricting fluid loss, defense against toxins and pathogen, and thermoregulation. Okay. Now, the composition of the blood, if you look at it, if you take out a sample of blood, this is the plasma component and this is your buffy coat, and this is where you have the whole erythrocyte. So, you take a sample of blood, you put it in a centrifuge and you get all these different components present in the blood. Now, coming to the different component. So, you have cellular component which constitute 45% of blood, which includes erythrocytes, which is the largest in number, the cells without any nucleus. It involves in transport of oxygen and helps in maintaining transport of carbon dioxide. Then you have a lesser number, which is the leukocytes or the white blood cells, which includes basophil, eosinophil, neutrophil, lymphocytes and monocytes, which is involved in the defense and immunity. Then you have a very smaller number which forms the platelet, which are involved in the blood clotting. Okay. So, these are the cell types. So, you can classify them as RBCs, WBCs or leukocytes and the platelets. Whereas, the major plasma component which constitute all different kind of salts and proteins includes sodium salt, potassium, calcium, magnesium, chloride and bicarbonate. It helps in osmotic balance, and this is the inspiration for developing medium. Cell culture medium is inspired by these compositions, pH buffering, regulation of membrane permeability. Then coming to the plasma protein, it is albumin, fibrinogen, and it helps in maintaining the osmotic balance, pH buffering, clotting and immunity. And the substances transported by blood includes glucose, fatty acids and vitamins, and waste products of metabolism, and exchange of respiratory gases and the hormones, and the solvents for carrying these substance, which is essentially water. It is a water based system which functions here, and this is the whole composition of the blood. Now, coming to the if you look at the whole thing, as I was telling you, is basically a fluid which is moving and it has lot of suspended particles. So, now having shown you this picture, now say for example, any of these particle numbers, say for example, these one increases or these one increases or these one increases, this will increase the viscosity of the blood, because the blood being a liquid in which there are particles in the form of cells suspended and smaller particle in the form of proteins and salts. So, if any number, any of this component number increases or decreases, the viscosity changes, and if the viscosity increases, the blood flow or changes. If the viscosity increases or decreases, the flow kinetics of the blood is getting influenced. Okay. Now, the part of the plasma which is made up of 90% water, as I was telling you, which is 10% protein, made up of minerals, protein, waste products, breakdown of protein, hormone, antibodies, albumin, clotting. So, there are a lot of places where you will see that the plasma is being stored, okay, and it has its own function. It has to be used for transfusion and several other things. So, obtaining plasma could be obtained by several ways, where you have to store the plasma, you separate out the blood and you take out the plasma. So, plasma composition, this is where the plasma composition comes into play. These are the different factors of clotting factors which are involved in it, and this is your plasma composition. This is the red blood cells and these are the platelet white cells. So, origin of the plasma protein is again what we have already covered, is liver. Apheresis, it is a process in which blood of a donor patient is passed through a medical device which separates one or more component of the blood and returns the remaining with or without extracorporeal treatment or replacement of separated components. Okay. Now, from here, this is another schematic showing the apheresis, how it happens. Apheresis donor, blood is drawn, blood is separated into component by centrifuge, needed components are collected in sterile bag, unused component are returned to the donor. The type of apheresis, plasma apheresis, plateloapheresis, RBC erythrophoresis, leukophoresis and photophoresis. These are the different components. So, now coming to, there are several biomarkers which are involved in the plasma proteins, and this is the kind of giving you an overall idea of the biomarker, and these biomarkers have different role to play in terms of detection of any kind of disease which are present in our body. Okay. So, now coming here, blood is drawn from the body, and this is where you are isolating the bone marrow stem cells. The stem cells which are involved in BMSCs are involved in formation of blood. I will come later about little bit more about this whole function of the BMSCs. Okay. So, formed element, journey of the BMSCs. So, the BMSCs, bone marrow stem cells, divide into erythroblast, which is forming RBCs, myeloblast, monoblast, lymphoblast, megakaryoblast. Megakaryoblast fate is forming platelets, whereas monoblast forming monocytes, lymphoblast forming lymphocytes, myeloblast forming three different fate: neutrophil, eosinophil, basophil. What is important here is this: there is a ratio which is being maintained. Between these BMSCs dividing into erythrocytes, white blood cells and platelets. and if this ratio shifts, then there are issues. If this ratio shifts in any direction, there will be issues. As a matter of fact, blood cancer is a situation when this ratio is kind of gets murkier. Okay. So, now coming back, these are the structure of the form elements: red blood cells, oxygen carrying, platelets for clotting, and these are your immune players, major players of immunity: lymphocytes, monocytes, eosinophil, basophil, and neutrophil. So, this is how the whole circuit kind of happens: multipotent stem cells, huge potential, and these progenitor are classified into two groups: common myeloid progenitor, common lymphoid progenitor. Then from the lymphoblast, they form T lymphocytes and B lymphocytes, which are forming the plasma cells. So, this is another route where T and B cells are happening, whereas common myeloid progenitor leads to megakaryoblast, which is forming platelets, whereas proerythroblast, erythroblast, and reticulocytes and red blood cells, whereas common myeloid progenitor leads to myeloblast, which is forming basophil, neutrophil, eosinophil, whereas promonocyte forming macrophages. So, these are the fate of the BMSCs which is undergoing the division. Then we come to the blood thinners, which are antiplatelet drug or anticoagulants. Natural blood thinner includes omega-3 fatty acids, which you can see all the different kind of natural blood thinners which are available in nature. Nature provides us. Now, coming back to the RBCs. Okay. This is how a RBC really looks like. These are the RBCs, erythrocytes. These are the monocytes which are multinucleated, neutrophil, lymphocyte with a large nucleus. So, what is hematocrit? Hematocrit is basically, so here you have the plasma fraction, here you have the solid fraction, which is 45% in case of anemia, where there is lack of blood, this is 30%. Polycythemia, this has increased to 70%, and during dehydration it becomes 70%. So, what is important to note is that this situation and this situation, the ratio I was telling you is kind of getting disturbed between the formed elements as compared with respect to, or the ratio of the form elements. Now, RBC formation factors: kidney releases erythropoietin, talk to you about it. Erythropoietin goes and hit on these bone marrow cells, and in the bone marrow these cells divide to form RBCs. Okay. So, all the factory is sitting in your bone marrow, and this EPO, this is an interesting molecule which is being used by several sportsmen. It is used as a drug for performance enhancement. Okay. So, as a matter of fact, one of the famous cyclists used this. Okay. Now, coming to the shape of the RBCs. So, this is how the shape of the RBCs is, and this is what I was telling you about the hemoglobin molecule, where iron is sitting at +2 oxidation state. So, this is the heme moiety. So, this is the globulin protein, the blue color what you see out here. This is the whole globulin protein. Okay. So, it has 4 units globulin protein, and at each unit there is one heme moiety sitting there. Okay. And each heme moiety is nothing but a porphyrin structure. If you remember this, I was drawing this structure for you. So, this was a way more complex structure, as you could see. So, iron is sitting there. So, this is the hemoglobin which is involved in carrying the oxygen, and it has beta polypeptide chains, which is the, and the alpha polypeptide chains, which is forming the globin molecule. Now, the oxygen carrying, so hemoglobin molecule is composed of 4 protein globin chains, is surrounding by central heme protein. These are the alpha chains, these are the beta chains. Again, these are the alpha chain, these are the beta chains. Okay. Here you have the heme group sitting out here. In most adult hemoglobin, there are 2 alpha chains and 2 beta chains, and this is where the iron is sitting, or coordination complex of the iron is sitting. Now, the binding kinetics is something like this on each unit. So, if you see, there are 4 oxygen which is getting bound to the hemoglobin, and this binding is kind of not a very strong or not a weak binding. When it is out here, it is loaded, and then at different partial vapor pressure of oxygen, it is being detached from this molecule. Now, what is very important to note is, if you look at the fetal hemoglobin and adult hemoglobin, you observe that the fetal hemoglobin have higher saturation. It means it carries more oxygen as compared to the adult hemoglobin. So, the reason being, the fetal hemoglobin is, as I told you, this is the adult hemoglobin. You have the alpha, beta, whereas out here you have alpha, delta, delta, alpha. So, this delta group, which is, so essentially there is a shift, like this, delta to beta as we become older. And if you look at this, this is the genetic variants which is happening, different genetic variants which comes up, and these are the chromosomes which are involved, chromosome 11, chromosome 16, chromosome 11, which are involved in generating different kind of hemoglobin structures. Okay. So, there are multiple variants of hemoglobins, what you can see, fetal liver, bone marrow, and yolk sac. I will leave it for you guys to go through it, and this is how the maternal connectivity happens between when it has to transport the blood and all other things. So, this is how the oxygen is being transported. Now, this is the maternal side, the hemoglobin present here and the hemoglobin present here. This hemoglobin has higher binding capacity. Because it has a higher binding capacity, it pulls the oxygen more towards it. So, that is how the ferrying of oxygen takes place. So, this is the hemoglobin molecule sitting inside an RBC and ferrying the oxygen. In the case of sickle cell anemia, which is a genetic disorder, the shape of the RBCs shifts like this, and because of this peculiar sickle shape, there are situations where these RBCs kind of clog the vessel. So, difference, the red blood cells are made in the spongy marrow inside the larger bones of the body. Bone marrow is always making new blood cells to replace old ones. Normal blood cells live about 120 days, mark this, this is important for you, 120 days in the bloodstream and then die. They carry oxygen, remove carbon dioxide. In sickle cell anemia, the abnormal sickle cell usually die after 10 to 20 days. So, this is quite a difference, and the bone marrow can't make new red blood cell fast enough to replace the dying ones. The mutation level, so this is where the sickle cell mutation taking place. GLU and valine. So, this is the mutant A which leads to the formation of the sickle cell anemia. Effect at the protein level, so this is the clumping which is happening because of the sickling of the cell. And at the organism level, this is the shift. So, a single mutation can lead to such a profound effect on the blood carrying capacity. Now, from here I will just introduce 2 terms before I close on this class. So, your immune system is divided into 2 parts, innate and adaptive, and the next class we will talk a little bit about it, and then we will move on to the next chapter. Thank you.